Imagine if the most effective cleanup system ever discovered wasn't a medication, a surgical procedure, or an expensive supplement. What if it was already built into your body, simply waiting for the right conditions to turn on? At this very moment, while you're watching this, your cells are slowly collecting damaged proteins, worn-out cell parts, and microscopic waste that continues to accumulate over the years. Most people never realize that the body has a natural process capable of clearing much of this cellular clutter. Today, you'll learn how that process works, what modern research has revealed, and what takes place inside your body when autophagy is activated at its highest potential. Every second of every day, your cells are incredibly busy. They're creating proteins, producing energy, responding to chemical signals, repairing DNA, and keeping countless biological processes running. But just like any machine that never stops operating, parts eventually wear out. Some proteins fold incorrectly and lose their proper function. Mitochondria, the tiny structures responsible for generating energy inside your cells, become damaged and less efficient. Harmful waste products also begin to build up. Unlike the trash you throw away each week, this cellular waste doesn't simply disappear on its own under normal conditions. As time passes, this buildup starts causing real problems. Damaged proteins can stick together, disrupting the normal function of the cell. Faulty mitochondria begin releasing excessive reactive oxygen species, increasing oxidative stress instead of supporting healthy energy production. Scientists have connected this growing cellular debris with faster aging, ongoing inflammation, and the early development of several degenerative diseases, including some of the most serious health conditions affecting people today. What many people don't know is that your body already has an advanced system specifically designed to solve this problem. It wasn't created in a laboratory or invented by modern medicine. It evolved naturally over hundreds of millions of years and exists inside nearly every cell of your body. For most of human history, people activated this system regularly without even realizing it, because food wasn't constantly available throughout the day. The real question is whether you're activating it today. In 2016, Japanese cell biologist Yoshinori Osumi received the Nobel Prize in Physiology or Medicine for his groundbreaking research on a process he had spent decades studying since the early 1990s: autophagy. The term comes from Greek, where auto means self and phagy means eating. Literally translated, it means self-eating. While that definition is technically correct, it barely captures how remarkable this biological process really is. Osumi and other researchers discovered that cells possess a highly organized, genetically controlled recycling system. When a cell identifies damaged or unnecessary components, it encloses them inside a specialized membrane called an autophagosome. This structure then transports the unwanted material to the lysosome, which functions as the cell's recycling center. There, these worn-out components are broken down into their basic building blocks so the cell can reuse those raw materials to create new, healthy structures. This isn't random self-destruction, it's a carefully regulated quality control system. The cell selectively removes only the components that are damaged or no longer useful, while preserving everything that continues to function properly. Before Osumi's discoveries fully changed scientific understanding, researchers believed autophagy was mainly an emergency survival response that cells used during periods of starvation. Further research revealed something much more significant. Autophagy is an ongoing maintenance system that supports healthy cells even under normal conditions. When functioning properly, it plays a major role in preventing the gradual cellular damage that contributes to aging and disease. The Nobel Prize recognized not just an interesting biological process, but one of the fundamental mechanisms that allows living cells to maintain their health over time. Your body constantly responds to internal signals, and one of the strongest influences on autophagy is whether nutrients are available, especially glucose and amino acids. After eating, particularly meals rich in carbohydrates or protein, insulin levels rise and activate a pathway known as mTOR, or mechanistic target of rapamycin. Think of mTOR as your body's growth switch. When it's active, Cells receive instructions to grow, build new proteins, and store nutrients. At the same time, one important process is intentionally slowed down: autophagy. This is why fasting has attracted so much scientific attention. When food intake stops, insulin levels begin to fall and mTOR activity decreases. As those growth signals fade, another pathway called AMPK becomes more active. AMPK acts as the cell's energy monitor. When energy becomes limited, it signals that resources need to be used more efficiently. One of AMPK's key actions is activating autophagy. Instead of focusing on growth, the cell shifts into repair and maintenance mode. Rather than investing energy into building new components, it begins removing damaged ones and recycling their materials. This isn't just a theoretical idea. Human and animal studies published in respected journals such as Nature, Cell, and the New England Journal of Medicine have documented these biological changes. What's especially interesting is that triggering this shift doesn't necessarily require prolonged starvation. Human biology is highly sensitive to changes in nutrient availability, so even relatively moderate fasting periods can begin moving the body toward greater autophagy. How strongly the process is activated depends on several factors, including fasting duration, overall metabolic health, and individual biology. Naturally, this leads to the question almost everyone asks when they first hear about autophagy. How long do you actually need to fast? The truth is more complex than many online videos suggest. There isn't one exact hour where a switch suddenly flips and autophagy instantly turns on. Instead, research shows that activation happens gradually. Some human studies have detected measurable increases in autophagy-related markers after approximately 12 to 16 hours without food. However, measurable changes aren't necessarily the same as maximum activation. Research involving muscle tissue samples and blood biomarkers, including studies published in Cell Metabolism, suggests that autophagy becomes much more pronounced somewhere between 24 and 48 hours of fasting in humans. Animal studies, which allow researchers to examine tissues more directly, consistently show particularly strong autophagy activity during fasting periods lasting roughly 24 to 72 hours. One influential study published in 2019 by researchers at the Salk Institute demonstrated that a 24-hour fast significantly increased autophagy within intestinal stem cells, which are essential for maintaining gut health and supporting tissue regeneration. Generally speaking, as fasting continues within safe physiological limits, autophagy tends to become more active. However, individual differences play a major role. Someone who exercises consistently, sleeps well, and maintains good insulin sensitivity may activate autophagy more efficiently than someone living with insulin resistance or metabolic dysfunction. Age is another important factor. As we grow older, the body's ability to perform autophagy naturally declines. This age-related reduction is one of the key reasons many scientists believe impaired autophagy contributes to the biological aging process itself. While a 16-hour fast is a practical place to begin, the stronger cellular recycling described in scientific research generally becomes much more noticeable during longer fasting periods. Now picture what's happening inside just one of your cells. And remember, your body contains roughly 37 trillion of them. When proteins become damaged or cellular parts wear out, the cell starts constructing a thin membrane around this unwanted material. That membrane gradually wraps itself completely around the debris, forming a double-layered structure known as an autophagosome. Under an electron microscope, it resembles a tiny bubble enclosing cellular waste. Once formed, the autophagosome travels through the cell until it merges with a lysosome, the cell's recycling center packed with powerful digestive enzymes. Inside, the damaged material is broken down into its most basic components, including amino acids, fatty acids, and nucleotides. Instead of being discarded, these valuable building blocks are released back into the cell, where they can be recycled to create new structures and support essential biological functions. This isn't an isolated event. It happens simultaneously in numerous locations within a single cell. while the same process is taking place across trillions of cells throughout your body. During an extended fast, the production of autophagosomes rises significantly. Research has shown that fasting enables cells to process and recycle far more damaged material than they typically do when nutrients are constantly available. This recycling serves a much greater purpose than simply keeping cells tidy. The amino acids recovered from old proteins help preserve vital tissues, build new proteins, and support organs that the body considers most important during periods without food, especially the brain and the heart. There's also a specialized form of autophagy called mitophagy, which focuses specifically on removing damaged mitochondria. This process is especially important because unhealthy mitochondria are linked to many of the diseases commonly associated with aging. When mitophagy is functioning efficiently, defective mitochondria are removed before they begin releasing excessive reactive oxygen species that promote oxidative stress and inflammation. Healthy mitochondria then multiply to replace the old ones, leaving the cell with a cleaner and more efficient energy-producing system. This is one reason longevity researchers are so interested in autophagy. The goal isn't simply removing damaged material. It's creating space for healthier, better-functioning cellular components to take their place. Autophagy is also far more selective than many people realize, and understanding what it targets helps explain why scientists consider it so medically important. One of its primary targets is misfolded proteins. These are proteins that fail to develop their proper three-dimensional structure after they're produced. Instead of simply becoming useless, they often become harmful. They stick together, forming toxic clusters that interfere with normal cellular function. These protein accumulations are one of the defining features of Alzheimer's disease. Amyloid plaques and tau tangles found in the brain develop, at least in part, because damaged proteins are no longer being cleared efficiently. Autophagy doesn't stop there. It also removes damaged organelles throughout the cell. Along with defective mitochondria, it can eliminate stressed endoplasmic reticulum, worn-out peroxisomes, and even harmful bacteria or other pathogens that manage to invade the inside of a cell. This defensive process is known as xenophagy. It allows cells to recognize invading microorganisms, surround them using the same recycling machinery employed for damaged cellular components, and destroy them before they can spread. Because of this ability, autophagy is considered an important part of the body's innate immune system. Another specialized form is called agrephagy, which specifically removes protein clumps that have already formed. This has attracted major attention in neurodegenerative disease research because brain cells have very limited ability to replace themselves once they're lost. Keeping these harmful protein aggregates under control is therefore especially important. A 2017 study published in Nature Communications found that increasing autophagy in mouse models of Parkinson's disease significantly reduced the buildup of alpha-synuclein, the protein responsible for forming the toxic Lewy bodies associated with the disease. The implication is remarkable. Autophagy isn't simply performing routine maintenance. When operating efficiently, it actively opposes several of the biological processes involved in some of today's most feared diseases. One of the fastest-growing areas of biomedical research is the relationship between autophagy and cancer, although it's also one of the most complicated. Early studies discovered that certain autophagy-related genes, particularly BECLIN-1, appear to act as tumor suppressors under specific conditions. Laboratory mice with reduced BECLIN-1 activity develop tumors at much higher rates than normal control animals. Researchers believe the explanation is fairly straightforward. When cells lose the ability to efficiently recycle damaged components, DNA damage accumulates, cellular stress increases, and the internal environment becomes more favorable for cancerous changes to develop. A comprehensive 2011 review published in Nature Reviews Cancer identified impaired autophagy as a recurring feature across numerous types of cancer in experimental models. The findings in Alzheimer's research are equally compelling. Scientists in the Nixon Laboratory at NYU, one of the leading research groups studying autophagy, discovered that this recycling system becomes severely disrupted in neurons affected by Alzheimer's disease. Instead of successfully completing the recycling process, autophagosomes accumulated inside nerve cells without fully breaking down their contents, leading to an increasing buildup of cellular waste. Later laboratory studies showed that restoring autophagy activity was associated with lower amyloid accumulation and improvements in several cognitive markers under controlled experimental conditions. These encouraging findings have made autophagy an important focus of multiple drug development programs that continue to be investigated in clinical trials. Research on aging tells a similarly consistent story. In one widely referenced study, genetically increasing autophagy activity in mice extended healthy lifespan by approximately 10 to 15% without requiring additional interventions. Studies using Caenorhabditis elegans, a tiny roundworm frequently used in longevity research, produced another important discovery. When scientists disabled the genes responsible for autophagy, the lifespan benefits normally seen with calorie restriction disappeared completely. That finding suggests something significant. At least part of the life-extending effects associated with eating less may depend on autophagy acting as the underlying biological mechanism. Across multiple experimental models, the evidence points in the same direction. Autophagy doesn't simply accompany healthy aging, it may be one of the essential processes that makes it possible. Ironically, one of the fastest ways to suppress autophagy is by eating too often. Every meal, especially one that raises insulin or strongly activates the mTOR pathway, sends a message to your cells to pause their internal cleanup program and shift their focus back toward growth, building, and nutrient storage. This response is completely normal after a meal. The challenge is that modern eating habits rarely give the body a chance to switch into its repair mode. Most people eat 3 main meals along with several snacks, stretching food intake across 14 to 16 waking hours. As a result, mTOR stays active for most of the day while autophagy remains largely turned off. A cellular recycling system that evolved over hundreds of millions of years often gets very little opportunity to do the job it was designed for. Some nutrients are especially effective at suppressing autophagy. One of the strongest is leucine, an amino acid found in high amounts in foods like meat, dairy products, and many protein supplements. That doesn't mean protein is unhealthy. Protein is essential. It simply means that consuming it, particularly in large quantities, reliably activates mTOR and signals the body to prioritize growth over cellular cleanup. Glucose has a similar effect by raising insulin levels. This is one reason some scientists who study autophagy question fasting methods that still include protein shakes or other calorie-containing foods. Even if total calorie intake is reduced, the molecular signals from those nutrients may significantly weaken the autophagy response. Another factor that's often overlooked is sleep. Research published in Nature Communications in 2019 showed that sleep deprivation reduces autophagy activity in the brain. That's especially concerning because neurons depend on autophagy to remove metabolic waste that accumulates throughout the day. At the same time, the brain relies on another waste removal process called the glymphatic system, which becomes most active during deep sleep. These two systems work together to help maintain healthy brain function, making quality sleep a critical part of your body's natural maintenance process. Chronic stress can also interfere. Persistently elevated cortisol levels have been shown to suppress pathways involved in autophagy. Ironically, many of the habits most strongly linked with poor health, constant eating, inadequate sleep, and long-term stress are the very same habits that keep your body's most powerful internal recycling system from operating efficiently. Based on current evidence, the most reliable strategy for encouraging autophagy is a combination of time-restricted eating and occasional longer fasts. For regular stimulation, limiting your daily eating window to around 8 to 10 hours, creating a nightly fast of roughly 14 to 16 hours, appears to produce measurable benefits when practiced consistently over time. For stronger activation, research most often associates fasting periods lasting 24 to 72 hours, performed occasionally such as once a month or every few months, depending on an individual's health and medical circumstances, with substantially greater autophagy activity.